Hello, everyone. This is Thomas Sir, CEO of Kinexum. Welcome to our July webinar, The Future of Type 1 Diabetes, Where Are We and Where Are We Going? A couple of housekeeping points first. We are starting out by muting everyone except the panelists for the sake of sound quality. Second, if you have a question along the way, please type it into the chat box or column, and the panelists will try to get to as many as we have time for. 3, we are recording this webinar. A YouTube video of the panel presentation and Q&A and a PDF version of the slides will be posted to our Kinexum website within a couple business days, and registrants will receive links by email when these materials are available. And 4, please stay tuned at kinexum.com for future webinars. One in September will be on aging biomarkers in regulatory development of anti-aging therapies, featuring Steve Horvath of UCLA, discoverer of the DNA methylation biological clock, legendary FDA official Bob Temple, and others to be announced. Today's panel features some of the field's top authorities in pharmacologic, advanced therapy, and device innovations for type 1 diabetes. We've included biographical sketches in the promotional materials. The panel will be moderated by Dr. J. Skyler of the University of Miami, a preeminent authority on diabetes and, among other things, former president of the American Diabetes Association. Jay, please take the mic.
Thank you, Thomas, and I'd like to welcome everybody to this webinar. As most people are probably aware, type 1 diabetes, we've made a lot of advances over the last numbers of years, but the type 1 diabetes exchange in the United States shows that we still do not have adequate glycemic control for patients with type 1 diabetes. Moreover, patients with type 1 diabetes get frustrated with the day-to-day impact on their lives of so many things they need to do in terms of balancing dietary things, exercise, insulin doses, and the like. And so they look forward to future developments. If I could have the next slide, Jennifer. What I think the ideal therapeutic goals in type 1 diabetes would be include 4 bubbles: prevention of immune destruction and preservation of beta cell mass, replacement or regeneration of beta cells, and then there's automated insulin delivery. And there's been a lot of progress in the latter over the last several years. And for that, we have Bruce Buckingham from Stanford, who will lead off our presentations and tell us a little bit of what the status of automated insulin delivery is. Bruce?
Thank you very much. So what I was going to do was present some of what I thought were some of the highlights from the ADA meeting, which showed some of the studies that are currently just ending or just finished, so it gives us a good update. So the next slide is the Metronics 780G. It's their advanced system, and this is from their pivotal study with 118 adults, 39 adolescents, and they came in with an A1C of 7.5, so they were in pretty good control. And then they had this crossover using a set point of 100 versus a set point of 120, and they were on each set point for 6 weeks. And if you can go to the next slide, I just wanted to show the type of results they got in terms of time and range. And if you look at the set point of 100, and you go across, we started everyone, because the engineer said to start at a 4-hour active insulin time, and they found as the study progressed that if you move that to 2 hours, and there were 29 of the total that were at this set point, they had a very good time in range, approaching almost 80% of 79%. And the interesting thing was, when you decrease the active insulin time, going from 4 hours down to 2 hours, the actual percent time in hypoglycemia was significantly lower, actually, and it did not increase when you shortened the active insulin time in the control setting. So I thought that was very encouraging to get close to 80% time in range with a pretty low incidence of hypoglycemia, and going from the 120 set point to 100 had about 2-3% improvement in time in range. Next slide. So this is using the same system, and it just gives a graphic presentation. I took this from Martin de Bock's talk from New Zealand, But you can see you always get this very nice improvement overnight. These graphs start at midnight and go to midnight the next day. So this is the overnight period where you see the big dip. And then as people sometimes forget to bolus, these are 14 to 21-year-olds, you don't get quite as good control during the day. Next slide. This is a study that we've been doing with the Omnipod system, and I think there's some unique things about it. You have the Dexcom G6, a no-cal sensor, talking directly to the pod, and the algorithm is actually on the pod. So as long as you're wearing these two, you're in closed loop, even without having a remote control device. And when you think about this, this pod's disposable and you change it every 3 days. And if you look at the 30% of admissions coming into the hospital for COVID have diabetes, and they have a much higher mortality rate. And if you keep them in good control, you decrease that mortality rate by 4 to 7-fold. So it's really important to control them early. And this is a type of a closed loop system that could potentially be used in the hospital for these people. It's something that hasn't been studied yet, but I think it's a unique opportunity. Next slide. This is the results from this study. I took the results from the 6 to 14-year-olds here, and they could choose their target. And over on the right side, you can see this, that about 60% chose a target of 110, but they had a 120, 130, 150, and the 150 was really used for exercise. And again, this is the modal day going from midnight to midnight, and a good improvement overnight. And if they use a 110 target, you can see that it was really nice in getting about 71% to keep their time in range with a very low incidence of hypoglycemia of a median of less than 1%. So very, very encouraging results. And next slide. This I'm presenting because we talk about control potentially from the onset of diabetes to try and preserve islet cells, prevent glucose toxicity. So this is a study being done by Roman Havorka, and it was presented by Charlotte Mutton, and they took children between 10 and 17 years of age, started closed loop within 21 days, and then we're measuring C-peptide at baseline and 6 and 12 months, and there's the study goes on to 24 months. And what's interesting, they have asked for an extension now to go to 4 years. And next slide, I keep pushing this slide here. But this is the group they've enrolled. They've completed enrollment. They have 50 in closed loop, roughly 51 and 46 in MDI. And their results are going to be released in the next, or available in the next 3 months for the 1-year data. And I think it's encouraging that they've asked to get an extension to study these subjects out to 4 years. So I think they're probably seeing some pretty reasonable results. And it's maybe the new way to manage diabetes is to really get them started on the closed loop system pretty early. Next slide. And I thought one of the really interesting things was they interviewed the people that were recruiting for this study. And there's a tendency to look for someone who's coming from technology, sometimes it's the engineer family, and recruit them into the study because they think they're going to be able to handle it well. And then there are some people that aren't as versed in technology, and you sort of worry about asking them to join a system like this. And it was actually the exact opposite. The technologically competent over-interacted with the system and made it harder to get better control. And the people that just accepted the technology did much better. And I think this points out a real healthcare bias that we really need to overcome in terms of how we look at people and recruit people into studies. And we really need to be very broad in approaching people. And all the studies that were presented really looked at taking people who were the adolescent missing boluses, the people with high A1Cs, and had a broad acceptance of people coming into the study. And those people did very well. Next slide. This is, I think, was very interesting. So this is from the Tandem Control IQ data that was presented last year. This is the 14 to 24-year-olds. And you can see that the big improvement in time and range, getting close to 70%, occurred on day one. It just occurs on implementing the system and getting that great control overnight and beginning to control the voltage. The variability is greater in that first 7 days, and it gets tighter as we did make some adjustments. So we are doing something, but you're getting people at about the same target. You aren't raising it. You're just getting some of the people that were a little outside improved, getting rid of some of the lows and some of the highs. But it happens very quickly. Next slide. So we looked at the group coming into the Tandem Control IQ who had A1Cs less than 6.5. And the interesting thing was they actually did better. And this is at nighttime. They had more time in range and certainly fewer lows at night in the blue down here at the bottom. And if you take the group that had a very high A1C above 8.5, they improved at nighttime about a 30% improvement in time and range, going from about 35 to about 65%. And if you go to the next slide, this shows the daytime. And again, during the daytime, the group that was in very tight control had less lows. And the group, this is the people that missed meal boluses, still had a significant improvement, about a 20% improvement in time and range, and almost had no lows. So I think a broad range people can be included in this. Sometimes the very compulsive people that are very good A1Cs are going to benefit at night, and everyone will benefit at night, and also the people that have high A1Cs. I'm almost done. Next slide. I think that's it. So I really think automated insulin delivery is becoming the standard, and Metronic pumps, Tandem pumps, and soon insulin pumps, which are the three manufacturers in the U.S., will all have automated insulin delivery when you buy a pump. For us, we're putting all of our subjects, actually 95%, go on CGM within a month after diagnosis, so that has become the standard. And now there are going to be two trials, the CLOUD, which was the Roman Hebrok study, and CLEVER, which is a U.S. study, using closed loop within the first 21 days of diagnosis. And I think as we begin to look at clinical trials, testing immune interventions, we're going to have to increasingly look at the confounding factor of having closed loop. And this may actually be an augmentation, and having the two working together maybe really will have a benefit for the islet cell. And I am done.
So I have a question for you, Bruce.
Yeah.
We hear a lot, and we did it during the virtual ADA, about the potential for dual hormonal automated delivery. And do you really pick up anything greater with dual hormonal? And what's the status of it, and your thoughts?
So it depends on which hormone you're talking about. I think if you had dual hormone with pramlintide co-administered with the insulin, that would be very interesting, and the data out of HADAR really looked pretty good for postprandial glucose values. And then with the glucagon, it allows the system to be more aggressive. So you can go from an average of, glucose of, 155 down into the high 130s to low 140 using bihormonal, because you have a safety mechanism at the back end of being more aggressive at a meal bolus. So I think for people that want really tight control, the bihormonal may have a role. And for people who have hypoglycemic risk, it may, or hypoglycemic unawareness, it may have a role there too. So I think we have to wait and see till we get a big outpatient pivotal study and see what it looks like. But they both have, I think, unique possibilities for improving things.
So one other question from Steve Gittleman, who wants you to comment on the time it takes for overnight to tighten up control. He says that often midnight to 3 a.m. is higher, but tending lower, and things are not great until 3 a.m. and then into breakfast, and wants to know if you need to alter behaviors such as the timing of the nighttime eating and the like in order to get it right overnight.
I think it it's a little bit dependent upon the algorithm you're using and what people are eating at night. But so the Tandem Control IQ is having the broader target of 80 to 160 coming in till midnight. And then when you put in your sleep hours, over 4 to 5 hours is lowering that target to 110 to 120. So it's partly an algorithm adjustment that's happening overnight with Tandem. When the Metronic is in the Insulin are to a target. They're to a set point, whether it's 100, 110, 120. So they're going to be trying to get there as soon as it can after a meal. So sometimes it's just the late absorption from a high fatty meal that may be keeping it up a bit. So I think you have to look at which system you're using.
Great. Thanks, Bruce. We're going to move on, and we're next going to take up immunotherapy. And for that, we have Chantal Mathieu from Leuven in Belgium. And Chantal also is the chair of and principal investigator of the European Union Consortium ENODIA, which is looking at immunotherapy across the EU. And I guess also you include the UK with you, even though they're not part of the EU anymore. I don't know where the UK stands in all this, but
don't go there, Jay. Okay, thank you very much, and thank you, Zen and friends at Kinexum for inviting a girl to the panel and a European to the panel.
Oh my gosh.
When Jay asked me to talk about immune therapy, I was tempted to list again in a very standard way what has worked, what hasn't worked, or what has partially worked. And then I thought, let me just list again why it is so difficult to arrest the progression of the process that causes type 1 diabetes. Let's not forget, we have this very long preclinical phase. We all have the picture of George Eisenbart with the long flat face, and then over months and years, functional beta cell going down. New evidence shows that this curve may be very, very difficult, very, very different from individual to individual. With some people even starting off lower with a functional beta cell mass, all the evidence coming in that perhaps the pancreas of people developing type 1 diabetes as a whole organ is also different. But then in some individuals, the curve going down in functional beta cell mass is very steep. In others, it's much flatter. So we're still learning a lot. And our problem is that this is a very, very difficult organ to access, because it is dealing with the beta cells, this very small mass, well hidden in the pancreas. And so this is one of the reasons why it is so difficult to get to an arrest of this disease. The fact that it affects young people, children, really raises the bar of safety when we are trying out interventions, especially immune interventions, because we are weighing efficacy and safety, and the threshold for allowing a therapy in a 57-year-old woman with arthritis is different than in a 3-year-old child. Animal models, we have the NOD mouse, but this is one of the fun sessions in a conference where we bash the NOD mouse. It is what it is, and it is very different when you look at the histology in the course of the disease and what have you. And so many people are saying that what we learned in the NOD mouse is a reason why we have gone astray in humans, because it seems to be a very different disease. I'm still a believer that the NOD mouse can teach us something, but indeed we need to have the proof in humans. And I already said the fact that this organ, the beta cell, is so difficult to access. For instance, biopsies, we cannot take a biopsy, or you can, but then in my country, the ethics committees would have problems. But also imaging is so difficult because it's such a small mass. And I really believe that biomarkers are needed, not only biomarkers of the progression of beta cell destruction, the progression of the immune attack, and you can say again, the antibodies we have learned is something, yes, but we need better, more accurate biomarkers. But we also need biomarkers of therapeutic efficacy, so that we can do our trials in a different way, that we can do shorter trials and have different endpoints. So next slide. In order not to make ourselves completely depressed after my talk, I have also listed what have we learned from the past 15, 20 years where we have done intervention trials in humans. And indeed, if we haven't yet prevented or stopped type 1 diabetes, it's not for lack of trying. What we have learned is that the immune system plays indeed a central role. We've learned also that the beta cell is not just a sitting duck and is not sitting there waiting for the immune system to be destroyed. I already said that the pancreas, the beta cells of people with type 1 diabetes are probably also different and behaving in a strange way. But we do know that if you give an individual with newly diagnosed type 1 a new bone marrow, like what has been done in Brazil, we can do something. And we do know that short-term immune modulation at the time of diabetes diagnosis, and now with the teplizumab study also in the months preceding diagnosis of type 1 diabetes, we can temporarily arrest the loss of functional beta cell mass. And so the next slide is a slide I stole from Jay and keep adapting, where we do have interventions that show that if you interfere with the immune system, you can freeze the loss of functional beta cell mass at least for a couple of months. We've shown that with bland immune suppression like cyclosporin. We showed it now with more subtle immune modulation like with anti-CD3, with more broad immune modulation like with low-dose anti-thymocyte globulin, and Steve Gittelman is on board, and so also learning from different doses of immune modulators is so important. So we have now evidence that indeed, if you interfere with the immune system, especially with T lymphocytes, you can arrest temporarily the functional beta cell mass loss. Next slide, please. Another important lesson, and again, when you make this statement, type 1 diabetes is a heterogeneous disease, clinicians start to roll their eyes because of course we know it's a heterogeneous disease. A child with a diagnosis at 3 years of age will behave clinically very different from a person who gets the diagnosis at 30 years of age. The loss in functional beta cell mass is different, the aggressiveness of the disease is different, the embedding of the disease is different. Some people only have type 1, others have all kinds of autoimmune diseases. So of course it's a heterogeneous disease. But the next slide, we're now also learning that when you look at the histology of pancreases of people who died shortly after diagnosis with type 1 diabetes in NPOT or in the EU NPOT that we have in Inodia, or in the biopsies in the studies that we have, it is obvious that also the infiltrate is different. And this is a work by Noel Morgan and Sarah Richardson, where, for instance, this observation that in young children with type 1 diabetes, you have this accumulation of B lymphocytes in the pancreas, in contrast to those who are older at the time of diagnosis. And so this may help us also in tailoring immune interventions. Next slide. And then another, again, I come back to this need of biomarkers, and because all the attack is happening in the pancreas, perhaps also in the draining lymph nodes, but everything we have is really blood, urine, stools, but so we need biomarkers in these peripheral tissues. Next slide. And so this is, and Jay alluded to it, oops, Europe has disappeared. But so this is Inodia, this is our our network, and the arrows go to the countries. So it is a public-private partnership between pharmaceutical companies, including Novo, Sanofi, Eli Lilly, GSK, Novartis, and academics spread throughout Europe, basic researchers and clinical researchers. Next slide, please. So what we do there is really look for novel biomarkers in a completely agnostic way. So this is, for instance, what we're doing now as an integrated analysis of the first 100, as we call them, newly diagnosed individuals. We have everything on these people. We have the clinical characteristics, genomics, transcriptomics, lipidomics, what have you. You can read it here. Everything goes into the same warehouse. And then with bioinformatics, we look for signatures. And so we do believe this is the way forward. Next slide, please. And then finally, and this is actually the immune therapy, the big challenge is indeed convincing clinicians of the benefits of an intervention, trying to arrest the progression of type 1 diabetes before the diagnosis or after the diagnosis. I mean, I also give talks on novel technologies, on the hybrid closed loops, on novel insulins and what have you. Let's not forget that clinicians out there really have now the impression that you can live with type 1 diabetes in a very good way, that with the pumps and the sensors and what have you, there's not really a need for interventions anymore. And this is a different world from 15 years ago. And so weighing the benefit and the potential harm that we do with immune intervention is really a delicate discussion we need to have in the first place with those living with type 1 diabetes in a family, for instance. And we believe that we need trial designs that allow us to test more drugs more rapidly. Next slide. And so, yeah, so this is the teplizumab study, but I'll not dwell on that. Next slide, please. So what we believe in Inodia is and this has been discussed with EMA, so with the European Medicines Agency, our FDA, that we need a master protocol where all the trials that are done in our consortium are built on. And so this is what has been approved by EMA, this master protocol. So all trials running in Inodia have this as the minimum minimorum. Next slide. They can do more measurements. For instance, here our low dose ATG dose finding study has more sampling in the beginning for pharmacokinetics, for toxicology, but it has the backbone of the Inodia protocol. So I believe there are reasons why type 1 diabetes is a hard disease to stop or to prevent. And there are important lessons learned. And we believe that with Inodia joining forces, not only academics, but also the pharmaceutical industry, novel trial design, and really going in depth to novel biomarkers will allow us to get to this disease with novel immune therapies.
Thanks, Chantal. What do you think is the possibility that you can get more and more people enrolled prior to the time of diagnosis? You know, the teplizumab study that you showed briefly, where you showed the published slide where there was a delay of the disease for 2 years, but at ADA, virtual ADA, last month, was shown to be at least 3 years. But it took a long time to enroll, and trying to find those people took screening nearly 150,000 people to enroll under 100 in the study. And so how can we do better at getting people identified earlier and enrolled potentially in studies earlier?
Again, my hopes are all on our biomarker discovery. If we can find signatures in peripheral blood, so in Inodia we have newly diagnosed, but also these unaffected family members that we screen, where we have all these things together in those with autoantibodies and those without. So I do hope that we will find signatures that allow in a sample of blood to identify people who progress. not only in family members, but also in the general population. And as you know, there's a parallel initiative where we're involved in Europe called the GPAT platform, funded by Helmsley, where Annette Ziegler is screening in the general population for people with a high genetic risk. Again, when you think of the effort this demands, it's just not feasible. So I think we will only be able to really make the needle move, Jay, when we find biomarkers that allow us to go out and find people in the general population without a high cost. And it may be a big challenge, and it may be we will have to focus on unaffected family members for now, but if already there we can find those that would benefit from our immune therapies, we have a foot in the door, and that together with better biomarkers should allow us to make a difference.
So Annette tells me that it costs €6 to do all the genetic screening on a sample obtained at birth.
Yes.
Should we not consider, since we screen at birth for many other diseases, should we not consider screening the entire population in the EU, the US, and Canada at birth for genetic risk of type 1 diabetes, and in those individuals follow them for the kind of markers you're talking about?
Yeah, I mean, that would be ideal, but I can tell you in Belgium, we're screening 100,000 neonates, and picking up a genetic risk is not all. You will pick up people who have a less than less than 5% risk of progressing to type 1 diabetes. And this is still a big group of people with a very low risk of type 1 diabetes. So as long as we only have genetic risk, this to me is too crude to really go out and say we're able to find people where it's warranted to do immune interventions. So I believe genetic together with some, proteome, metabolome, a small interfering RNA, we hope to find it. But that will be needed, Jay, so that at least you can talk to people and say, well, you have a risk of 1 in 10 or 1 in 3 instead of less than 1 in 20.
Yeah. So Robert Spiegel wants to know what you meant by transient effect on that slide that summarized previous interventions.
Yeah, so this is very interesting because if you look at all of our interventions in the newly diagnosed people, it's almost like you revisit again and again and again the effect. Every time you see, it was also shown with the anti-IL-21, with the Novo study now at ADA, you see this effect for 3-6 months, and then things start happening again. And all of our interventions seem to like freeze the immune attack against the beta cell for 3 months, 6 months, some 9 months, and then it starts again. So yeah, this is the transient effect.
Yeah. Well, we're going to need to move on, but thanks, Chantal, for a great review. As most people know, we can, in fact, reverse type 1 diabetes with pancreatic or islet cell transplantation. And pancreatic transplantation is usually reserved for people who are also undergoing a simultaneous kidney transplant. And islet cell transplantation for people who have recurrent severe hypoglycemia resulting in multiple emergency room visits. Well, let's assume that Chantal and colleagues can solve the immune problem, not only of the disease, but of rejection. And we could, in fact, do islet or pancreas transplantations. The sad commentary is that in the United States, each year, according to the UNIS, the Organ Procurement Agency, only 1,200 pancreases potentially become available. We're not going to reverse a lot of type 1 diabetes with 1,200 pancreases. We need something more. We need an unlimited supply of cells that can be used. And to address where we stand in using stem cells, both to understand the disease and potentially to treat it, we have with us Matthias Hebrok, who's the director of the Diabetes Center at the University of California in San Francisco and one of the world's foremost people in dealing with stem cell biology as it relates to type 1 diabetes. Matthias?
Yes, hello everyone, and thanks for having me. Thanks for the introduction, Jay. Can I have the next slide, please? I briefly have to show you this. My university actually requires me to show this, but I think we can move on to the next one. Just to set the stage again, I know that almost everyone on this call is very well aware of these numbers, but I think they're still shocking. Right now in 2015, so already 5 years ago, in the world, according to the International Diabetes Federation, we had around 450 million people who were diagnosed with diabetes, and of course that includes type 1 and type 2 diabetes. But starting from 15 going to 2040, which is now only 20 years ago, there's going to be 642. And certain areas of the world going to have a dramatic increase. For example, in Southeast Asia, where you go from 78 million or so to approximately 140. That's almost a doubling. So there is a real urgency for us not only to understand type 1 diabetes, of course, and type 2 diabetes, but also to come up with ways to mitigate the disease and hopefully to actually really fully cure it. Next slide, please. So if you strip everything away from both type 1 and type 2 diabetes, of course, there's an immune component in type 1, but at the end of the day, you're ending up with the depletion of beta cells, either functional exhaustion and then a loss of the beta cells, or mediated through the immune system, a real destruction of the beta cells. And therefore, this is the cell type that is missing in people who have been diagnosed with diabetes and are suffering from the disease. And therefore, at least in theory, as Jay just alluded to, us being able to regenerate this cell type at will is something that should help patients with diabetes. Now, on the right side, I have depicted not just the beta cell, but the islet. And that is something that we call a niche or a neighborhood. And as it turns out, actually now the differentiation conditions that we are using allow us to not only just generate these beta cells that produce insulin shown in green, but also several of the other cell types that are normally found within the human islet. And we actually do believe that being able to regenerate the full complement of an islet is going to make these cells even more potent, more robust, and more functional. So as Jay again alluded to in his brief introduction, if I can have the next slide, the proof of principle experiment has been done. What I mean by that is that here you have a patient actually, and it was done by my colleagues at the University of California, San Francisco, where you have this patient and he was on an insulin pump. And then, as you can see here in red, this person actually obtained an islet transplantation. And as we have seen this before by Bruce Buckingham when he described the new development of the automatic delivery systems that are really fantastic. But this is a biological way of doing this. And you can see how the range in which the glucose levels are now going to be controlled is very, very tight. Essentially, in many of the patients, is very similar, if not identical, to what you would find in a patient who's not suffering from diabetes. So what that means is the proof of principle experiment exists. If we can take or regenerate human islets, transplant them into patients, we will be able to regulate the glucose levels in these patients in a way that they are, for all intents and purposes, are cured. Have the next slide, please. Now, I don't have time to go and talk to you about all the different ways of how you potentially can do this, but suffice it to say that my lab, as well as several others around the country and actually around the world, have taken this on as a challenge. And over the last probably 20 or so years, we have identified ways to either start with, on the left side, and what we call a human embryonic stem cell. Now, that is a cell that is derived from the inner cell mass of a human embryo. But we also can take what is shown just below that, it's called an iPS cell, and use pluripotent stem cell. And that is a really interesting and very potent cell because it can actually be derived from somatic cells. So from cells that we have in our body, the skin or the blood, using specific kind of modifications, we can turn them into a state that is very similar to this embryonic, fully potent stem cell state. And then by using something that we call directed differentiation, essentially what we're doing is we're giving these cells cues that they would normally receive during the differentiation in the embryo to turn them into a number of different, what we call progenitors. The one I'm showing here is a bipotential progenitor. We already have at that point reached the stage of the pancreas. As many of you know, the pancreas not only generates the islets, actually the islets are only 2 to 5% of the pancreas. The vast majority are duct cells and acinar cells. So we have to then use other signals, and I've shown several of those here, to take them away from what we would call the exocrine part of the pancreas towards the islet, the endocrine part. Now, in the beginning, we are generating immature beta cells, and that was the state of the field, the whole field, probably 4 or 5 years ago. But over the last several years, we have learned to, again, by modulating TGF-beta signaling or resizing the clusters, or by just going in and reclustering and modifying the size of these clusters, to be able to generate clusters that now actually consist of mature beta cells. And as I just mentioned to you before, we also find other endocrine cell types in these clusters, including alpha cells and delta cells. So the promise of all of that is that in theory, we should be able to generate as many cells as we want from pristine cell populations, ES cells or iPS cells, and those can then be used, upon expansion, of course, to be transplanted into patients. If we can have the next slide. Again, I do not have time to give you all of the details, but I just want to briefly show you what we can now do with these cells. If you just focus your attention on the right side, this is the gold standard. It's a human islet. And what we're looking forward here is the insulin secretion based or stimulated via glucose. And what you can see is that there's low levels of insulin secretion in the absence of glucose. If you go to 20 millimolar glucose, so a real bolus, a strong bolus of glucose, you have a nice spike. But even in some of the human islets, you have this first phase, but not necessarily a sustained second phase. Some islets show this, others don't. You can further activate insulin secretion by giving exendin-4, GLP-1 analog. And then, of course, if you give potassium chloride, you have a spike of activity. Now, if you contrast that to what you show on the left side, that is an islet structure that we derived several years ago, which I would call an immature cluster. So these are these immature beta cells. They're clearly beta cells. They express many of the markers that we find in human beta cells. But if you just look at the beginning, the first part, when we go to 20 millimolar glucose, you see that the level of insulin secretion is very low, not even perceptible. Now in the middle, we have EBCs, and these EBCs are what we call the enriched beta cell clusters. And those now have a very nice peak of first phase insulin secretion. Again, the second phase, as we see in some of the islets, is still lacking, and we're working on optimizing that. But you see a nice response to exendin-4 as well as to potassium chloride. So if you take all of this together, and we have done many analysis, if you guys have an interest in this, you can actually look this up in the paper we published last year in Nature Cell Biology. So what we did is we kicked the tires, and we really asked the question, how close are these cells that we're now generating under cell culture conditions to those that we find in our body in the human islets? And the answer is that they are somewhere in the vicinity between 85 and 92% identical by transcriptome analysis to those that we have in our bodies. So there's still a little bit of room to grow, but we are very close in generating cells that are more or less equivalent to human beta cells. Now the next slide is my last slide, and it indicates that it's not just us, of course, who is able to do this. There are a number of groups around the world, actually, that are now further activating and optimizing some of the conditions that we and others have described. But I think what I'm trying to say is that we are now in a position where the information exists to generate, hopefully at will, beta cells and actually full complements of islets that can be used for transplantation purposes. Now, the last point I want to make, and I don't have a slide for this, but I want to just briefly mention this, is that everything that we have done was done with embryonic stem cells, and iPS cells are working as well. But there was a revolution, of course, all of you guys are aware of, in terms of gene editing through the CRISPR system that was developed over the last 5 or so years. And this system is just unbelievably powerful. We now can gene edit our embryonic stem cells or iPS lines, again, almost at will. And what this allows us to do is to make cells, as we're working on, more robust, for example, to withstand the assault of hypoxia or the immune assault upon transplantation. So by being able to modulate the genomic content of our cells by activating or deactivating specific factors at will, we, I think, will be able to not only generate islet cells, but cells that are more robust, more functional, and therefore hopefully more able and for a longer extended period of time, help patients suffering from diabetes. That's all I had.
Well, thank you, Matthias. I notice that a couple of these article titles that you have on this last slide talk about generating beta cells. But you've made the point, and I think it's that it's an important one, that beta cells alone probably are not what we need. We need islets. Yes. Because the paracrine effects of the islet are so important, particularly if we want to use these like islet transplantation is currently used for people with recurrent severe hypoglycemia. I'd like you to comment about that.
Yeah, you're absolutely right. I mean, we're learning more and more that, as I said, the beta cells are living in a niche, in a neighborhood, and that at least we think now there's a trifecta of 3 different cell types that work together, possibly actually being controlled by somatostatin produced by the delta cells that actually has an effect on both the alpha cells and the beta cells. We do know that the alpha and the beta cells also talk to each other, and therefore being able to generate the full complement is something that I think is desirable. Now, a lot of these papers do not talk about this. We actually mentioned this in our paper. One of the reasons is that the beta cells that we're generating, first of all, there's more markers. It's easier to ascertain their functional activities. The beta cells look quite good in terms of their maturity states, the way we are doing this. Now, the alpha cells and delta cells that we have in our cultures under cell culture conditions are clearly present. They appear to be slightly immature, so they don't behave exactly the way they should behave. Upon transplantation in a surrogate animal, an immune-compromised mouse, for example, that seems to resolve over time. So we do think that we're generating at least the progenitor cells that have the ability to turn into full complements. We now have, and this last point I want to make, us and Doug Melton just had a paper, as well did we, recently, where we showed that by modifying the conditions, we can also go away from just mainly making beta cells to actually the majority of cells we're now generating are alpha cells, and we're working on delta cells as well. So I think we're going to be learning a lot, and we're going to be identifying molecules and factors that would help the maturation of these cell types as well.
One of our listeners wants to know how invasive would be the procedure of putting these cells into human beings?
Yeah, this is of course is a great question. As many of you know, what we're doing right now still is most of the time with islet transplantation is we're putting into the portal vein, and then the islets are lodged into the sinusoids of the liver. That is not necessarily the best place to put them. For the stem cells, there's a number of different ways to look at this. There are still considerations in terms of the safety of using these kind of cells. So early on, and there's companies who are working on this, they might have to be encapsulated just to protect the person who receives the transplant. I think that this is moving into the background, to be honest with you. I think the safety of these cells is going to be adequate soon, and going to be equivalent to human islets, which means we would be able to just use naked cells without any kind of protection. Now, at that point, if I'm correct, and we're working on this, that we, for example, could stave off hypoxia, which is one of the assaults that any kind of transplanted cell receives upon transplantation. If you find ways to block the hypoxic cascade in beta cells, that would essentially allow likely for those cells to be a little bit in a dormant state for like a week or two and then become fully functional. That would allow for many of them to survive, and that would allow us to put them into places where vascularization is not as high as it is in the liver, for example, subcutaneously or intramuscularly.
One of our participants wants to know, are you concerned about premature senescence with iPS cells?
Yeah, that's a great question. Well, first of all, I think my job is to be concerned about everything. So I wake up being concerned, I go to bed being concerned. So yes, of course, we worry about these things as well. iPS cells, we are working with them. A lot of people are working with them. They are still not quite as good as human embryonic stem cells, at least in our hands. The human embryonic stem cells is the gold standard. It's the real deal. And we still continue to use them because they seem to be more robust. Having said this, for iPS, what we've learned is that it actually depends on the age of the donor, because there are mutations that occur in the mitochondria in people who have reached my age, for example, over 50. And therefore, it is possible, and people assume that it will be the case, that you have to screen a number of iPS lines that you generate from each patient to find the one that really has the best properties, not only in terms of just being fully redifferentiated into a stem cell, but also in terms of the properties in being able to give rise to the endoderm that then later on gives rise to the pancreas and the beta cells. So yes, we worry about these things. I don't think that these are insurmountable problems. I think this is just something that over time we will have to address, and we will be able to do this.
Isn't it not also true that the problem with iPS cells is you have to de-differentiate them first and then redifferentiate them in the other direction, whereas with embryonic cells, they only go in one direction?
That's exactly the point. So what you have to do is you have to do it twice, more or less. You first take a cell that is already differentiated, is set in its ways, and you have to force it really hard to put it into a state that is a de-differentiated state. And that's what I mean. In some cases, that even if you get 95% there, that might not be enough. You might have to go to 99%. And therefore, you might have to establish several lines from each patient to really get to the line that is as de-differentiated as possible and therefore as malleable as possible. Absolutely correct.
Well, thanks. Appreciate your excellent review of this. We're going to move on to our last formal presentation before we open it up for group questions more. And please use the chat box, folks, for sending in questions for any of the speakers, and I'm happy to relay them. And our last speaker is, it says on the slide, Alexander Fleming. We know him as Zan Fleming. Zan, as you may know, was the founder of Kinexum, who's hosting this conference. But I knew Zan when he was the diabetes group leader at the FDA. And my first interaction with him when I went to see him in his office at the FDA was particularly intriguing to me because he had better furniture than I'd ever seen the government issue anywhere. And it turns out Zan likes to make his own furniture. And he did, in fact, make the furniture in his office himself and got government permission to bring it in. And that's why I was so impressed with the furniture in his office, because it was not government issue. Anyway, Zan is going to try to summarize where we stand from a regulatory and a industry standpoint in trying to find the solutions for type 1 diabetes. Zan?
Well, Jay, what an introduction. And what an honor it is to be part of this extended family gathering of friends and colleagues who have been for many decades seeking a transformative approach, or approaches, for type 1 diabetes. You are responsible for my passion for type 1 diabetes, going back to that eventful meeting we had in Rockville, where you came to talk about the diabetes prevention trial, DPT1. Of course, it was in its early stages then, but that was a landmark trial in the history of type 1 diabetes development. It was a Herculean effort on your part and your colleagues' part, on a par with DCCT. And it, specifically, for those who do not know, evaluated the autoantigen insulin. Even back in the early 90s or before, insulin had been identified as, in fact, near the root cause of type 1 autoimmunity. And so, Jay, you were instrumental in leading that trial, which also led to the formation of TrialNet, which you led, and has been so important in supporting other type 1D interventional trials. So it's been a real honor to be your comrade in arms. And it is a daunting challenge for me, an old regulatory guy, to be representing the industry on this distinguished panel. It's daunting in part because obviously there is no greater diversity of industry approaches in all the therapeutic development approaches that are being aimed at type 1 diabetes. Think about it. From small molecules to cell therapies to complex device systems, type 1 diabetes is unique in all of human medicine for the variety of technologies that are being aimed at achieving what we might call a practical cure. Come back to that term in a moment. But it is quite amazing to think that only perhaps in the cancer field are so many different modalities being used to target a practical cure for type 1 diabetes. Now, I have had the privilege of being involved in the development of all of these different categories. And so I'm glad to speak on behalf of multiple sub-industries, though I do so with trepidation, and knowing that there are many experts on the phone who have far greater knowledge than I do of the details. Next slide. Well, we are here today for a very simple reason, and that is because injected insulin therapy is among one of the most difficult and hazardous drugs used to treat any chronic condition today. And the result is that serious hypoglycemia is a threat that hangs like a sword of Damocles over people who have type 1 diabetes. And that ultimately is why we need a technology that will give us a very reliable, easy to use means of achieving normal glycemic control. And we could call that a practical cure, if you wish. Next slide. Well, we have covered, going from right or left to right, we've covered closed loop device systems, which are maturing into really amazing technologies to improve glycemic control. Bruce has covered that well. Matthias has talked about cell therapies and the great progress that is being made there. We have a wonderful presentation from Chantal about the various interventions that have been used to preserve, typically, beta cell function, and usually people with new onset type 1 diabetes, and to prevent it, as we recently have seen with teplizumab and its prevention trial. There are even some small molecules that are being repurposed that have immunomodulatory effects that are, I think, very promising. But there are actually a number of things that we aren't going to cover today, and I just want to put them in as placeholders. First of all, there are so-called smart insulins, and if you had a smart insulin that was truly smart, that in itself would take care of providing a very easy to use, safe product. There are patches that release insulin in response to ambient glucose control that are under development. There's even a small molecule that is directly targeting the reduction of hypoglycemia. And there have actually been several attempts in that direction that could, with a small pill, make the therapeutic index of insulin safer, or improve the therapeutic index of insulin. And then I want to come back to Matthias, who is really an amazing leader in the field of islet cell biology and helping to actually manufacture islets. I'm hoping that once he has finished with that project, he will actually come up with a way to generate islets in the pancreas. And I believe this is possible. We'll come back to that in a moment. But just to say, we're not going to talk about islet regeneration therapies, which is yet another area for development. Next slide. I have to be very general here in comparing and contrasting the various parameters that apply to these different approaches. Automated insulin delivery systems are here. They're already very successful. They're making major strides in improving glycemic control in people with type 1 diabetes. But they have a typically short life cycle. There will be many different iterations that occur over the next years. We'll see smaller, more convenient to use systems. The manufacturing is relatively straightforward, though certainly it involves a lot of sophistication. And it doesn't take a lot of clinical data to get a AID product on the market relative to what it takes for, let's say, immune agents. And the regulatory burden is likewise relatively low. It's not to say that it's absolutely low. There certainly is a lot of work, time, and money involved. But relative to complicated clinical trials that have to be done with immune agents, it's relatively easy. Cell therapies fall somewhat in the middle, and that we have a pretty good idea of what clinical data would be necessary and sufficient to approve these products. The regulatory burden is relatively low as long as for now there is not a safety concern, and that is addressed, as Matthias mentioned, by encapsulation. But these products have been under development for many, many years, and so that has not been an easy pathway. And Chantal covered well the field of immune agents that have been tried since going back to the 80s, maybe even before then. And therefore, the life cycle is very long. The risks are very high. The manufacturing may be moderate to high, depending on the product. And there has to be a lot of clinical data to show not only effectiveness, but safety. And the regulatory burden is consequently relatively high. Next slide. Chantal, I think, summarized very well the issues in developing type 1 agents to address type 1 diabetes autoimmunity. The preclinical models are imperfect. The large clinical trials that are required have to be pretty long, 1 to 2 years with long-term follow-up. And in contrast to these other approaches, the expectations for showing the effect size of a product that modulates the immune system and the secondary outcome results are less clear. Obviously, no therapy has been approved yet, and it continues to be a question as to what does the expert community need in terms of what would be sufficient to say that a product is safe and effective. Next slide. All I want to do here with this complicated slide is simply remind everyone, and many of you need no reminder at all, some of you actually participated in getting these data, that just a little bit of beta cell function preserved in people with new onset diabetes leverages major clinical outcome benefits. And that's the reason that the bar should be relatively low and what that effect size should be in assessing immunotherapies. So we have very good reason to believe that if we intervene at any point and can show some improvement in endogenous insulin secretion, that can ultimately reflect a long-term clinical benefit. Next slide. Now, the point I want to leave you with is that all the modalities that we've been talking about are complementary. And actually, Bruce made this point when he mentioned the Hobarth data earlier on, is that, for example, AID can improve conditions for islet restoration. And if we do that in the presence of autoimmunity, it gives us a better chance if we are looking at the performance of an immune intervention of seeing long-term benefit. And it goes without saying that if Matthias gives us naked islets to put in the body somewhere, if we have an approach to control the autoimmunity of type 1 diabetes, that is going to make a big difference. And so all of these therapies are complementary, and we should not look at them as so much competitors, but as different regiments of the army that are aiming to fight a common foe. With that, let me just, as a vision slide, take next slide, remember the Joslin medalist. And this is why I have hope that Matthias and his colleagues can eventually find ways of returning endogenous insulin secretion in the pancreas itself by finding the reason that human beings do not have strong regenerative islet capacity. And this is just, as many of you know, a snapshot of islets in people that had had diabetes for 50 years or more on insulin, showing some evidence of islets being formed, some evidence of insulin being made, and in a number of these cases, C-peptide can actually be measured. So we know that there is a low level of islet regeneration or beta cell replication that's going on even in older people with type 1 diabetes. And if we can find a way, a safe way, of controlling that autoimmune attack, then we have the prospects that people, type 1 diabetes, not only those with new onset type 1, but those with well-established type 1 diabetes may ultimately benefit by a partial or, who knows, complete restoration of endogenous insulin secretion. Next slide. Just a little advertisement. I'm privileged to be a member of the EASD ADA Technological Working Group, and we just put out a paper on digital apps. But next slide, the next paper that is coming out is on automated insulin delivery devices, and so stay tuned for that. Well, again, it's a great pleasure to be part of this panel, and thank you, Jay. Back to you.
Thanks, Ian. I appreciate your presentation. You know, I like you raised an interesting issue with the Joslin medalists, and you know, it leads to a question which I'll pose to Chantal first, but maybe Matthias wants to get in and comment as well. And that is, when we have islets in type 1 diabetes, are the beta cells destroyed, or are they just sleeping when they lose their function? And can they potentially be reawakened if they're just sleeping? And either with you know, some form of you and your colleagues have demonstrated that in vitro, at least, GLP-1s have the potential of helping prevent apoptosis of beta cells. You want to comment, and then maybe Matthias as well?
Yeah, I mean, indeed, one of my pet topics and your pet topics is the fact that this beta cell is not an innocent victim, but that harnessing it with agents like GLP-1 receptor agonists, or probably there are better agents out there that can make stronger beta cells, would allow us to give synergistic effects with immune modulation. Now, your question, what happens in the pancreas? Again, I think we need to forget these images we have from the NOD mouse, where we see massive infiltration, destroying beta cells very rapidly. When you look at the images coming from NPOT, first of all, it seems a very patchy process where there are islets where beta cells are still there at the time of diagnosis. So where for sure adding something that can make stronger beta cells will help. And then when you go, and again, Zan showed it to the gold medalists, people who have had diabetes for a long time, beta cells are there. Now, also, Jay, when you, again, sleeping beta cells, Kevin Harold showed that there is a heterogeneity also in the beta cells in islets where you have infiltration. So I believe there are some sleeping beta cells, some dedifferentiated beta cells, but there's also a lot of killed beta cells. So I think it's a mixed phenomenon where you have apoptotic beta cells, but also where you have dedifferentiated beta cells. But I don't believe that they're all sleeping. And I don't believe all of them can be resuscitated.
Matthias, anything to add?
Sure. How much time do we have? I mean, it's a really amazing phenomenon, isn't it, right? And what Zan said and what Chantal said, of course, is true, but there's a number of different issues here. So the first one is that the easiest way would be just to proliferate the cells that we have left. But as Chantal said, some of the beta cells that exist in type 1 diabetes likely are not just innocent bystanders. So even if you make more of those, the immune system is revved up to essentially attack these cells. There are people like Andy Stewart, who is actively trying to essentially break the brakes in the beta cells to have an increase in proliferation using harmine and TGF modulation and these kind of things. So if we could essentially get this specific to beta cells, and there are others out there who are using adenoviruses, Mark McKay, Marcus Grompe, so we might be able to have a tailored therapy that just hits the beta cells and erodes the blocks for proliferation, at least for a certain amount of time, and get more. But again, the issue then is that we're doing this in the context of an immune system that is just ready to take these cells and take them out. So we would have to then also understand why that is the case. What is different in the beta cell in a type 1 diabetic patient compared to controls? And is there something that we can block, for example, the recognition by the immune system of these beta cells? All of these things are being investigated, and the human stem cell system allows us to do this. So we, for example, among others as well, we're taking at HLA molecules, and we're seeing that there's actually, it seems to be a protection against the human immune system. Long story short, it's very, very complex. I agree with what Kevin showed. And we had shown this before in a different context, that beta cells can go dormant or de-differentiate. That essentially possibly provides them a little bit of protection against the physiological need to make a lot of insulin, but also possibly against the immune system. But I don't think that these protections are enough over a long period of time in type 1 diabetic patients. I think at the end, the beta cells are being recognized and they're being destroyed. So can we do this? Nothing we cannot do, but we have to really fully understand all of the aspects on the immune side and the beta cell side.
So, and when you're dealing on the immune side, when you're replacing cells like with yours or others, one has to consider both allo rejection of those cells as well as the recurrent autoimmunity. And I think that makes it even more complex because there may be different pathways. It's been shown that you get recurrent autoimmunity even in the face of sufficient immunotherapy to prevent rejection.
Yeah, absolutely. I mean, that's unfortunately for type 1 diabetic patients, it's the double whammy, right? I mean, not only to have the allo, but then we have the auto, which means even if you make the beta cell, then that particular cell type is being recognized as foreign. It's daunting.
Zan, Philip Holm wants to know if you have transient, less than 5-year improvements in C-peptide, is that going to be an acceptable regulatory outcome for effectiveness?
Yes, I don't think there's any doubt about C-peptide being the necessary but not alone sufficient endpoint for showing efficacy or effectiveness ultimately. The question will be, how much do you need to see? And that will take an expert judgment. And Phil, you may be called to serve on the FDA advisory committee, so we want to start coaching you in the right direction. Don't be a herd grader here. We've got to get a lot of different choices out in the market so we can pursue Jay's dream of combinational therapy, which I think we all understand that there's not going to be a silver therapeutic bullet, even for the immune side of type 1 diabetes. We're going to need multiple agents that, like in cancer therapy, can ultimately achieve a cure.
Is there going to be a difference, Chantal, between regulatory agencies in the US and the EU and elsewhere in the world?
I hope so. I hope that the EMA will be progressive and that they will understand that to show evidence beyond C-peptide in the first year of diabetes treatment will be very difficult for any intervention. Let me say what I mean. I mean, our people, as Bruce showed, we're putting them on sensors when they are newly diagnosed. We're giving them intensive insulin therapy. And in this first year, I mean, the number of severe hypos we see is very small. So, you know, to bring out this advantage of a small difference in C-peptide in the first year after diagnosis will be a challenge. So you will need to go 2, 3, 4 years. Imagine the nightmare of the study. So I hope that just stimulated C-peptide will be enough for EMA, because otherwise we are in trouble.
And Jay, I just want to chime in and say amen to Chantal's remarks. People should recognize that C-peptide is not a biomarker. It's the better way of measuring insulin itself. Type 1 diabetes is a near absolute deficiency of insulin. It's a hormonal deficiency state. And in my former FDA division, we always approved hormonal therapies by simply showing that you could normalize the levels. And therefore, we should look at it that way. It drives me crazy when people say, well, C-peptide is just a surrogate marker. It's not enough to show that you're making a difference. I think, to Chantal's point, we have to have a reasonable standard that will encourage the development of multiple approaches that can be evaluated in the real world.
And you know, Chantal raised something else too in that comment, that everybody is going to be on sensors.
Yeah.
And Bruce, if they're already on sensors as well as automated insulin delivery, is that really going to complicate the ability to detect things other than beta cell function, which we can easily measure with C-peptide? But is it going to complicate looking at things like hypoglycemia and glucose control in order to get things done?
Well, I think things are going to be better. I think actually you have a better way of measuring those outcomes. So, I mean, if you're getting on a population less than 1% hypoglycemia, less than 70, it's hard to see a huge significant improvement, but that's a very good goal. And then you're looking at the top end. And so you're, instead of maybe a range of 70 to 180, you may want to range closer to 70 to 140, and you can begin to bring these targets down, which shows some endogenous insulin production if you have a much tighter range like that. And that's what you showed with the islet transplant, that very clamping of glycemic variability and getting nighttime, daytime a lot better. If you have some C-peptide to help with the meals, it makes a huge difference. So I think we may have to, I think you're going to need CGM to really be an outcome measure also when you begin to look at these things.
Yeah, it's very strange, if I may, Jay, it's very strange. I mean, I agree that variability expressed as time in range is a surrogate for functional beta cell mass, but C-peptide is the reflection of functional beta cell mass. So we go in circles. I mean, C-peptide is it. Stimulated C-peptide shows you what is happening in the pancreas.
Absolutely. So Chantal, Steve Gittleman wants to know where you stand in the EU and the EMA in trying to get children into immunotherapy trials. He points out that we've become hung up a bit in the FDA world in them wanting adult-only trials first, where because you have a better chance of showing benefit of immunotherapy in younger people who have a more rapid decline of beta cell function, that you maybe have a difficult time showing prospective benefit before moving into children. And how does it stand in the EU and with the EMA is what he would like to ask.
Yeah, here the situation is actually even stranger in that the regulatory authorities, there's always one country where you need to submit your dossier first. So I was with a company 1.5 years ago, and they used the Belgian authorities to submit their dossier for advice. And the authorities and EMA push the companies to go to lower ages. They all make them propose a PIP, a pediatric intervention plan. And so the company wanted to start at 6, and the Belgian authorities and then later EMA said, no, you need to start at 2. So it's the opposite. Here they are really pushing to, in type 1 diabetes, they acknowledge that it's a disease of children with a very specific course in children. So they push to very young ages where even the pharmaceutical companies feel uneasy.
Right. Johnny Ludvigson says if the treatment is very easy for the patient, then it may be justified even if the C-peptide preservation is just 10 to 20% for some years. But with a dangerous treatment, you might look at it differently. I think that's a fair comment that most of us would agree with. Paul Feldman wants to know any role for an impact of GLP-1 receptor agonists and SGLT inhibitors for type 1 diabetes patients. That's adjunctive therapy, and we did not include that in the discussions today. Chantal is an expert on that and may want to comment just briefly, but I think, Zan, maybe we need another seminar in type 1 diabetes where we include adjunctive therapies as one thing, regenerative therapies as another, and some of the things that we didn't get to cover today. And Thomas and Zan can think about whether or not we ought to do that. But Chantal, do you want to comment on that?
Yeah, I mean, so walking away from the beta cells, so SGLT2 inhibitors, as you know, again in Europe too, have been approved, and clinicians are using them, but are using them with great caution in hand-picked patients because there is the increased risk of diabetic ketoacidosis. Again, as you saw in our real-world evidence, we are at 4% compared to 1 to 1.5% in non-SGLT2 type 1 people. So it is a risk you need to tolerate, but those people who are using them see great advantage in A1C, in weight, and especially Bruce, in this flattening of the curve, the better time in range. GLP-1 receptor agonists, to me, is even more interesting because, as you know, this was demonstrated with liraglutide many years ago. Novo didn't like it because of the increased hypo risk we saw. But as type 1 specialists know, this was just a reflection of not down titrating the insulin rapidly enough, giving you cumulative increases in hypo. But now there's again a growing interest in retesting GLP-1 receptor agonists with the better products we have. So I do hope we can convince companies with interesting compounds to go there, because I think for weight, the GLP-1 receptor agonists are very interesting in people with type 1. And for preserving beta cells, as you saw in the recent publication by the Danes, there is a possibility of doing something for preserving functional beta cell mass.
Great, thanks. Thomas, I'm going to throw it back to you because we're running just towards the end of time, and I know you have a couple closing remarks. And I want to thank all the panel for their willingness to participate in this and respond to my requests. So I got 100% successful response, so I felt pretty good about that. Anyway,
Thank
Thomas.
you, Jay.
Terrific. Thank you so much, Jay, and all of our panelists, and also to the audience for making this a terrific webinar. I'm loath to bring it to a close. I know that we advertised this to be over at 12:30, but in any case, this webcast will be posted to our Kinexum website, and links will be sent to you by early next week. And also important, you may receive a survey that will provide us with feedback about how to make these webinars better. Please fill them out if you do. And with that, I would thank the speakers and attendees and wish you all a good day and a good weekend, great weekend.